Setting up for transcription in mammals is regulated by many cis-regulatory elements, including core promoter and promoter proximal elements that are located near the transcription start sites of genes. Core promoters combined with general transcription factors are sufficient to direct transcription initiation, but generally have low basal activity. Other important cis-regulatory modules are localized in DNA regions that are distant from the transcription start sites. These include enhancers, silencers, insulators, and tethering elements. Among this constellation of elements, enhancers and their associated transcription factors have a leading role in the initiation of gene transcription. An enhancer localized in a DNA region distant from the promoter of a gene can have a very large effect on gene transcription, with some genes undergoing up to 100-fold increased transcription due to an activated enhancer. Enhancers are regions of the genome that are major gene regulatory elements. Enhancers control cell type-specific gene transcription programs, most often by looping through long distances to come in physical proximity with the promoters of their target genes. While there are hundreds of thousands of enhancer DNA regions, for a particular type of tissue, only specific enhancers are brought into proximity with the promoters that they regulate. In a study of brain cortical neurons, 24,937 loops were found, bringing enhancers to their target promoters. Multiple enhancers, each often at tens or hundreds of thousands of nucleotides distant from their target genes, loop to their target gene promoters and can coordinate with each other to control transcription of their common target gene. The schematic illustration in this section shows an enhancer looping around to come into close physical proximity with the promoter of a target gene. The loop is stabilized by a dimer of a connector protein, with one member of the dimer anchored to its binding motif on the enhancer and the other member anchored to its binding motif on the promoter. Several cell function-specific transcription factors generally bind to specific motifs on an enhancer, and a small combination of these enhancer-bound transcription factors, when brought close to a promoter by a DNA loop, govern level of transcription of the target gene. Mediator communicates regulatory signals from enhancer DNA-bound transcription factors directly to the RNA polymerase II enzyme bound to the promoter. Enhancers, when active, are generally transcribed from both strands of DNA with RNA polymerases acting in two different directions, producing two enhancer RNAs as illustrated in the figure. 18. An inactive enhancer may be bound by an inactive transcription factor. Phosphorylation of the transcription factor may activate it, and that activated transcription factor may then activate the enhancer to which it is bound. An activated enhancer begins transcription of its RNA before activating transcription of messenger RNA from its target gene.